Hello. Yes, thank you for the introduction. It's great to be here to discuss what we've been working on at Vizgen, especially at this time for spatial biology. As mentioned in a session previous, we really start to see this convergence of what was previously maybe disparate technologies, highly multiplexed genomics-based technologies that now are starting to be able to be combined with spatial. So you can start to have this highly multiplexed measurement where you really understand what's happening across the tissue, but now include the spatial information in that type of measurement. So if we look at the history of genomics types of technologies, so taking it from that perspective, if we look at bulk sequencing, of course we learned a lot there about what are the genes, how are they expressed across different tissues. With single cell sequencing, we learned even more. We learned how are those genes expressed among different cells in order to achieve different functional states in those cells. But we're still missing this critical aspect of biology, which is this intricate spatial organization. These cells aren't just working in isolated droplets, but rather they're spatially organized across the tissue in order to create this overall functioning, or in some cases in disease tissue, dysfunctioning biological system. So being able to map the single cell information back to exactly where those cells came from gives this new way of looking into biological systems, this new way of really understanding what's happening, whether it's a healthy tissue or a diseased tissue. So what I'll be discussing today is Vizgen's MERSCOPE platform that aims to do exactly this that we just launched earlier this year. So MERFISH combines MERFISH, originally published back in 2015 from Zhao Zhuang's lab, that combines the power of single molecule FISH quantification with the error-robust barcoding scheme to achieve very highly multiplexed image-based spatial transcriptomics measurement with the easy-to-use platform solution that streamlines the whole workflow all the way through custom gene panel design, sample prep, imaging, and downstream analysis. With this platform, you can now run panels of hundreds of genes, so really relevant to understanding the complexity of the biological system across large tissue areas up to a square centimeter of tissue, all with single molecule resolution across that full square centimeter with high accuracy and sensitivity, as I'll be discussing, with no need to extract the molecules and put them into a sequencer. It's all direct in situ detection. And now, although MERFISH existed as a technology previously, now it's much easier for new labs to start to adopt this technology using the MERSCOPE solution that streamlines the full process. With this, since our launch earlier this year, we're already starting to see a lot of adoption among a whole range of different areas of biology, neuroscience, oncology. But since any real biological question, or nearly any biological question has a spatial component, we do imagine that there's, as with any these predecessor genomics technologies, there's this whole range of research applications that are just waiting to be untapped with new applications of this technology to different biological questions. So today I'll focus the talk around three main aspects. First, I'll introduce MERFISH technology, so you know exactly how this instrumentation and how this platform is working, how we're doing this very highly multiplexed spatial transcriptomics measurement. From there, I'll briefly discuss the workflow, how it looks to run a sample through the platform, all the way from gene panel design through getting the data out the other side. And then I'll finish up by discussing how we're continuing to expand the capabilities of this platform. In particular, how we're expanding the different sample types that it can work on, from fresh frozen to FFPE tissues as well. So I'll share how some of that data looks as we work to launch that capability later this summer. So MERFISH stands for Multiplexed Error-Robust Fluorescence in Situ Hybridization. It's a technology originally invented and published back in 2015, coming from Zhao Zhuang's lab, where there's this growing interest, both shown in the rapidly growing number of publications around the technology, especially in the last year or two, and the growing number of institutions using the platform. But as the name suggests, from FISH, it's built around existing technologies, of course. It builds on what came before it. In this case, single molecule FISH. So in a single molecule FISH measurement, you can design fluorescently labeled oligos that bind specifically to a target transcript. So once you stain with these fluorescent oligos, you can visualize them with a high resolution fluorescence microscope in order to count exactly where each copy of this transcript is across the sample. So it's very quantitative. It's direct counting of these transcripts in situ. But it's limited by the number of distinct molecules you're able to count simultaneously within the same tissue slice, because it's fluorescence-based. With traditional fluorescence measurements, you maybe measure 3 or 5, sometimes 7 distinct colors within an imaging round. But to really understand the biology, there's, for example, in the brain, there's hundreds of different subtypes of inhibitory and excitatory neurons. To really characterize that, you have to measure a representative number of genes, hundreds or thousands of genes. So to build single molecule fish up into something that's that highly multiplexed, MERFISH introduces this binary encoding scheme. So instead of reading the genes out one at a time, each gene is assigned a unique, error-robust binary barcode, as shown here. And then once we have the genes assigned to the barcode, then we're able to read them out through sequential rounds of imaging, where wherever there's a 1 in the barcode, it's going to show up as a fluorescent spot in the image. Wherever there's a 0, it remains dark. So for example, here we just lay out the sequential imaging process. In the first image, we're reading out the first bit in the barcode. So any gene that was assigned a barcode with a 1 in this first bit position shows up as that single molecule fish spot, while the ones with 0 remain dark. So we image this, record the image, extinguish the signal, and then restain for the second bit in the barcode. Now we have a new pattern of single molecule fish spots corresponding with all the genes that were assigned a barcode with 1 in the second bit position. Again, we extinguish and then continue through the process to read out the full binary barcode. Then from these images that we acquire, we can go back and look at the pattern of brightness and darkness across the sample, identify the barcode, and then since we know which gene was assigned to which barcode, this enables this very highly multiplexed spatial transcriptomics measurement. So with this, since it's imaging-based, you can really measure biological samples across whole tissue slices up to a square centimeter of tissue with single molecule resolution across that whole tissue slice. So that enables analyzing the molecular composition of the sample all the way from single molecule to subcellular, to neighborhoods, to tissue microenvironments, all the way up to the macro tissue level scale. And you have this information across the whole tissue, not just at specific points that you've selected across the tissue. But for any measurement, it has to be both highly quantitative and give high quality data. So here, with the MERFISH output, we can compare that to bulk sequencing. We find very good correlation with bulk sequencing all the way to the lowest expressing genes. Here you can see in this mouse brain measurement, we have strong correlation all the way from high expressors, nearly 1,000 FPKM, down to the lowest expressors, even less than 0.1 FPKM. So nearly 4 orders of magnitude of quantitative gene expression. But additionally, for transcriptomics, since transcriptomics is a relatively sparse signal in the first place, a lot of very functionally relevant genes are expressed at just a few tens of copies per cell. The ability to detect a large fraction of those tens of copies is really critical to understanding what's happening in that biological tissue. This comes in the data in the terms of detection efficiency, or what fraction of expressed transcripts are you actually able to detect. So here, since it's direct in situ measurement, we don't have to extract the RNA molecules, put them through molecular biology to amplify the signal. Rather, we just fix the tissue, hybridize to stain it, and then read out these binary barcodes. So it's a very efficient process because each extra step that you have, whether it's extracting the RNA, amplifying it, you have the chance of losing some of the molecules in the process. So here, when this comparison with MERFISH, with a technology that involves that type of extraction, when we plot out the expression of one of the genes, opioid receptor delta 1, we see this very rich spatial organization that's invisible when you have much lower detection efficiency. The other advantage here is that with direct detection, you have no loss in accuracy because when you extract molecules often, you don't know exactly where that molecule came from. But here you can see exactly where this opioid receptor delta 1 is expressed within these individual cells. And in this comparison, we find that there's nearly 70-fold more transcripts detected with the MERFISH measurement than with this DNA extraction-based technology. So from the technology, now I'll briefly go through the workflow or how it looks to run a sample through the MERSCOPE platform. Since each biological question has its unique set of genes that are most relevant to it, we offer entirely custom gene panels. So to help with that, we have a gene panel design tool where you can put the genes that you'd like to target. It gives immediate feedback about whether those are most suitable for the MERFISH measurement, lets you refine it and finalize to create your gene panel for your biological question that then you can order, and Vizgen produces that in a ready-to-use format. Once you receive that gene panel aliquot and you have your tissue slice ready, or tissue block ready, you can take a slice of that tissue block, place it onto the MERSCOPE slide, fix and permeabilize it to hold everything in place, and then place a drop of this gene panel mixture onto the sample so that the probes that encode these binary barcodes can bind to their respective RNA targets. After that binding, then we have a gel embedding and clearing process, because across the whole range of different biological tissues, there's a whole source of different autofluorescence background. So to help mitigate that and make sure we give the cleanest possible images and the highest data quality, we embed the sample into a polyacrylamide gel and then clear away the proteins and lipids so that any of these extra sources of autofluorescence are then cleared away, so we can actually cleanly image these single molecules across whole tissues, across the whole variety of different sample types. After that gel embedding and clearing process, the sample is ready to go into the flow chamber, and then along with the reagent cartridge, they get loaded into the MERSCOPE instrument. The MERSCOPE instrument automatically goes through the sequential imaging process to read out these binary barcodes. It acquires all of the images and then automatically processes them into the biologically useful output. Here, for this type of measurement, the biological useful output is first the primary output of a spatial transcriptomics measurement, which is a list of detected transcripts. In a single measurement at the highest capacity, we can almost get 1 billion transcripts out of a centimeter squared tissue slice in a single measurement. But from this, it just lists out the XYZ coordinates of each detected transcript and the gene identity. In addition to that, we also perform cell segmentation so that you can immediately out of the platform do single cell analysis. So we segment the boundary of each of the cells, and with that segmentation, we can then go back and count how many transcripts of each of the different genes fall within each of those cells, and in this way, construct a cell-by-gene matrix. So this cell-by-gene matrix is just like what you'd get from single-cell sequencing experiment. It fits into the same sorts of tools like SROT or ScanPy in order to cluster cells, identify different cell types across your sample. But now we have all the spatial information, so we can map those cell types back to their spatial coordinates. Additionally, since these are very large files, it's typically, even with a lot of bioinformatics expertise, it's not trivial to plot out plots with billions of points in them, so we also have interactive visualization software that lets you see all of the transcripts, all of the cells, and all of the images acquired during your measurement, so you can start to explore what's actually happening across your sample. So some examples of where MERFISH has been used are first this maybe flagship MERFISH paper back in 2018 published from Zhaowei Zhuang's lab, where they used MERFISH to profile the mouse hypothalamus. So here, from the MERFISH measurement, they were able to characterize all of these subtypes of inhibitory and excitatory neurons with very good agreement with what they saw from single-cell sequencing. In some cases, even identifying cell types that were invisible to single-cell sequencing. With the spatial information, you're able to map those back, see how these cell types are varying across this region of the brain. And they even overlaid that with early activity genes, CFOS, in order to figure out which of these neuronal subtypes were activated during different mouse behaviors that were implicated in this area of the brain, like parenting, aggression, or mating behaviors. Another example for MERFISH technology is this more recent example in oncology. So here, they want to understand the microenvironment within the glioblastoma sample. In particular, they wanted to understand the interactions between macrophages and some of these glioblastoma cells, in particular these mesenchymal-like cells. So they did MERFISH, characterized the different cell types across the glioblastoma slice, and they all actually found enrichment of macrophages in the neighborhood of these mesenchymal-like cells, which helped supported their overall hypothesis that the macrophages were helping to induce some sort of transition within these cells. But to help with the whole range of applications that we see coming from MERFISH technology, within Vizgen, we have been demonstrating MERFISH on a whole variety of different tissue types, including mouse and human tissues. Here we're looking at fresh frozen tissues, but of course the other common sample prep type is FFP. So along with the fresh frozen protocol that is currently part of the platform, we're also in the next few months releasing our FFP procedures that I'll go through as well. Because each of these different sample types has their own unique advantages. Of course, with fresh frozen, it's nice to have a fresh frozen tissue block because you can maybe use part of that block for single cell sequencing, use another part of that block for spatial. But it's not the sort of universally perfect sample type. It's not the clinical standard in many cases where protocols aren't available to process tissues from certain samples into FFP blocks. But it's also sometimes biohazardous if there's an infectious agent within the tissue. On the other hand, FFPE tissue blocks have their own set of advantages. Since they're fixed early on, it helps preserve the tissue morphology. But at the same time, they're more prone to degradation, especially with the typical ways that they're stored at room temperature. So since it's an RNA measurement, it's really critical to have ways of measuring the RNA. So we've been working to optimize the protocol for some of these more fragmented or degraded samples. And that's what I'll be discussing over the course of the next few slides. So to show how it works, we can look at this mouse brain, because with mouse brain, it's very easy to compare data quality across different conditions. Here, using our FFP protocol, you can see that with the MERFISH measurement, we're able to measure the gene expression across the whole tissue with this single molecule resolution, just as with the fresh frozen measurement. And when we look at marker genes from the panel, we find very good correspondence with the established Allen Brain Atlas. But to farther validate this data, we also did co-clustering, where we have one mouse brain from fresh frozen sample and one mouse brain from a FFP sample, mix the cells together and cluster them together. And here, from this clustering, you can see that they're very well intermixed. So here, these clusters, should be a video here too. Okay, yeah, great. So here you can see the two mouse brain samples, the FFP and the fresh frozen. And you can see from the UMAP that these two distinct sample preparation types are very well intermixed in the UMAP representation without any extra batch correction, indicating that there's very high level of correspondence between these two distinct sample prep methods. To further validate that, of course, we can look at the correlation with bulk sequencing, which is similar to what we see in the fresh frozen case, correlation between replicates, highly reproducible measurement, and then correlation between our fresh frozen and FFP protocols. So then beyond mouse brain, we find similar data quality where across a whole variety of different human, both healthy and disease sample types, we find very good correspondence with bulk sequencing and very high quality data. So in particular, in addition to being able to do the RNA measurement, it's also compatible with RNA plus protein. So here, in addition to this panel of 347 genes, in this case, we also measured 3 different proteins, pan-CK, SMA, and E-cadherin. On the left, you see the protein distribution. On the right, you see the RNA distribution. And you can see all the way down to the single cell level. Now we can quantify both small panels of proteins plus large panels of RNA molecules simultaneously within the same FFP tissue slice. This really starts to reveal some of the intricacies that exist in a lot of these different oncology samples. There's a lot of really rich spatial organization. I think this is one of the most diverse samples that we've looked at so far, this FFP human ovarian cancer. Just from these 8 genes, you can already start to get an idea of the richness across the sample. But then using our visualization tool, we can zoom in to the sample and really start to see the microenvironment across the tumor. Here we're looking at 8, but then we can toggle on all of the genes and you can start to see all of these stromal cells across the tumor with hundreds of transcripts detected within each of the cells, which is more than enough to really characterize the cell type distribution across that sample. A lot of the other FFP samples we looked at are similarly rich, all the way from human skin, human tonsil, to a variety of different cancers. And some of our early adopters are starting to already present on some of the FFP work, in particular Compugen, and then Nir Ha-Cohen's group with the Broad are starting to use the FFP data to really explore the neighborhoods correspond it with their single-cell sequencing data to validate these different cellular neighborhoods that they've detected exist from single-cell sequencing across the tumor. And then they're using MERFISH to build upon that previous work. But overall, just in the last few minutes that I have, I don't want to take too much of your time from lunch. We're very excited to have launched the MERSCOPE platform at the beginning of this year, and really excited to see the number of initial applications or initial installs and initial biological applications that our early users are pursuing. And we're planning to continue that launch by expanding it into the UK and Europe within the next month or two. If you're interested in seeing what MERFISH data looks like, we have a couple of public data sets posted on our website, both this liver data set and mouse brain data set, and we'll be soon posting some FFPE data as well. But with that, I'll leave a few minutes for some questions. But as Vizgen is rapidly growing with this initial commercial launch, along with the interest in helping people run these type of measurements, we're also growing as a company. So with that, we have lots of open job positions. So if you're interested in helping to push forward the next generation of spatial transcriptomics, we'd be happy to talk as well.
Okay, before we break for lunch, does anybody have any questions for George at all? No? Okay. Well, we are running pretty much bang on time, so where can we find you if we have any?
I'll be at our booth
okay.
in the room for the next hour or so,
okay.
if you have additional questions.
Okay, great. All right, it just leaves me to wrap up this session then. Thank you for attending. If you'll just join me in thanking the speakers again for this session, and if there is any one-to-ones, you can make your way to the vendors' booths now. Thank you. Cheers.